Loviniski, who is a professor of chemistry, research director at the French National Research Council, and director of the Institute of Analytical and Physical Chemistry in Pau, southern France. He graduated as a chemical engineer from the Warsaw University of Technology, from which he obtained subsequently his PhD. He was postdoctoral fellow of the Max Planck Society in Germany, research assistant at the University of Antwerp in Belgium.
Essentiality and importance. So, the control over trace metals is a central question in physiology of any living organisms. So the metals can be acquired from nutrition, can be acquired from the environment, and how they are acquired. So that's a very important question. How they are acquired, how they are stored, how they are distributed. So we will discuss the mechanisms, and we will discuss the question of how analytical chemistry can help us to decipher these mechanisms. Well, those of you who are biochemists, they know it by heart. There is a gene, the gene codes for an enzyme. The enzyme produces metabolites, and some of these metabolites complex metals. So they are ligands complexing metals. So these mechanisms are extremely important, not only to understand the importance of metals, but especially to understand how these metals are important. So my lecture, I will discuss two major topics. One is a pure analytical chemistry. It's about how to describe a biological system in terms of trace metals. And the second is how to decode a biological process on the basis of information we obtain from advanced tools of analytical chemistry. So what is to describe a biological system in terms of trace metals is to learn the concentrations of all trace elements in the cell, is to define their precise isotopic ratios, because isotopic ratios, isotope composition, I don't think it's me, so it must have happened something to my presentation. Yes. So isotope ratios, why they are important? Because they evolve during the process of evolutionary or physiological fractionation. Then it's important to disclose the chemical speciation. So what are the forms of these trace elements, how they are complex, what are the ligands. Then we can go beyond the chemical speciation, whether there is importance of conformations of different biomolecules, whether there is importance of their stereochemistry, and finally it's the question of localization. So what is the distribution of these species in a tissue, in a cell, or in a subcellular compartments? So that's analytical chemistry. That's the core of my research, but we are involved in the full image of trace elements in single particles. So if you have single particles, nanoparticles, for instance, you can introduce them into a plasma. A nanoparticle takes more time to ionize. We are talking about microseconds here, but we will get specific signals for particles that are not present in the continuum of your sample. So ICP-MS, that's why this technique is important. It has become really a trigger for technology-driven research on trace metals in the context of tremendous advances in biology. So I think that the key event in metallomics was the introduction of this technique. Or hardly any biologist or biochemist heard about into the context of omics. Omics, the copist has already heard about. So the omics, well, it started all with genomics 30 years ago with this famous paper, or two papers in year 2000 to start the new millennium. on the sequencing of a human genome. Holistic approach in terms of global trace elements content of a biological system. We are used to the reductionist approach, which has been referred as bioinorganic chemistry. So it's studying the structure and function of individual metal complexes with individual ligands. But neither a metal or a bioligand is present alone in a biological system. So all the elements and many bioligands are present together, and they may interact. And the second important point, if you do something in the lab, it doesn't mean that nature has done it in the same way. So we are interested not in vitro metallomics, but in vivo metallomics. You want to approach a living system. And what metallomics change the perception of trace element information. It has become global, species specific, and embedded in the contents of other omics, so proteomics and metabolomics. So there were two concepts that were extremely similar, that were born about the year 2000. One, well, it's metallomics and ionomics, but basically they are all about the history of metallomics. So when you see the term in front of the title of this conference, that we know where it came from. So there are two major approaches to metallomics. One is just total element concentrations. So the information we get is the set of concentrations of all trace elements in a sample, and then we have to correlate these sets of concentrations with different genomes of bacteria, yeast, plants, or collections of epidemiologically characterized samples. So it's a correlative approach. Analytical chemistry gives you the set of concentrations, and then you've got other conditions, you make correlations. But you can go one step beyond that, and this step beyond is that you consider not the elements, but you consider the species these trace elements exist in a sample. And this is what we call speciation analytical approach. And speciation is about understanding what the metal complexes are. Understanding what the metal complexes are is the determination, the identification of ligands, and then imagining what enzymes could have produced these ligands, and finding the genes that were at the origin of the synthesis of these enzymes. So the classical trace elements metallomics that was born in plant chemistry at Purdue University, Professor David Salt, that was about studying social network of metal ions. So on one hand, we had ICP-MS, multi-element information, and on the other hand, we had large collections of plant mutants. So if you can imagine Arabidopsis, 4,000 different mutants, you analyze each of these mutants for the trace element contents, and then you can correlate the presence or absence of a particular gene, as you can see here. So on the x-axis, you've got trace elements and the concentrations. So you get such a picture for each individual mutant. Once you've got it, then you need to correlate this for the whole collection of mutants, and you will discover genes that are related to a particularly high or particularly low concentration of a given element. And this approach, I mean the correlation of the trace metal concentration pattern and the large set of biological or environmental information we used to study the correlation of trace elements with COVID-19 severity markers. So we could do it with our Russian friends. At that time, I was a visiting scientist at Sechenov University in Moscow. So as you can see, that there's a gradual decrease. If there is a decrease of trace elements, of important trace elements such as calcium, iron, selenium, zinc, then there is an increase of COVID-19 severity markers. So it means that the trace element concentration patterns are extremely important. So the paper was published a couple of years ago. It has a really quite nice impact, really underlining the importance of the global trace element analysis. Sorry? I think now, have you heard me correctly before? Yes? So maybe that, if I speak like that? So it was okay like that? Okay, thank you very much. So that's the importance of a trace element concentration pattern. So now let's come to more sophisticated and to the second things and to the second level of information that goes beyond the total trace element concentrations that that concerns the metal species. So why they are important? So I will just summarize the background of the importance of trace elements in biology. So metal ions bind to ligands via interactions that are often strong and selective. The ligands impart their own functionality and can tune properties of the overall complex, and these properties are unique from those of the individual ligand. Measurement, it's seconds. Here we've got the chromatographic run, so it takes about an hour. But within 1 hour, you get a huge amount of information on metal species in biology. What can you use in terms of the separation technique? Well, it can be, of course, chromatography, but it can be electrophoresis. It can be gel electrophoresis, but then you need a laser ablation to see the presence of different elements in different electrophoretic bands. So you've got plenty of the techniques. Well, with Joanna, we published 20 years ago a book on that, because all these techniques we developed at that time in our laboratory. So that was really an arsenal of tools for speciation studies. There is a problem with that. The problem is that there are more species in a biological, more metal species. I know there are biologists in this audience, so when I use the term speciation, it's not biology. The term speciation is chemistry. Speciation means different complexes of metal elements with different ligands. So there are more of them than the resolution of chromatography. So if you use one chromatographic mechanism, then you cut off a fraction. You can separate this fraction by a different separation mechanism, size exclusion in this case. Then you've got another fraction, another separation mechanism, and even after the fifth fraction, you still have many species. So you can get about 5,000 or something like that of detected metal complexes. So the resolution here is clearly not enough, because we are unable to separate all the species, metal species, the richness of a biological, of nature produced, the diversity of metal species by ICP-MS. So about the year 2010, we had in the lab an idea to change this paradigm. So to replace ICP-MS with the metal-specific information by a softer ionization technique that produced us the mass, molecular mass of the whole species, not only of the element. So it means that the challenge was shifted from the chromatographic peak capacity to the peak capacity in a mass spectrum, from the atomic pattern of ICP-MS to the molecular pattern of electrospray MS. So we have now, and this is important because mass chromatography has a resolution maybe of 100 species. Mass spectrometry can resolve thousands, if not millions. I will show an example later on. So this new paradigm, a single peak in a mass spectrum per metal compound, requires that all the metal or metalloid species can be ionized, because we need an ion. Why we need an ion? Because only ions can be separated in electrical or magnetic electric or magnetic field by mass spectrometry, and these ions have to be separated, and for that we need resolution. So here comes the cavalry. Extremely high resolution mass spectrometers. So these pieces of equipment are worth, I would say, €1 million, $1 million, or something like that a piece. But with Fourier transform, ion cyclotron resonance, mass spectrometry, we can get to resolutions of about that are more than million. Well, this means that we get the molecular mass with the accuracy of the 4th digit after the comma, and if we get it with such a resolution, it gives us the empiric formula. And if it's the 4th place after the comma, this means basically that we can resolve about 10,000 species without a particular problem. We've got, Orbitrap MS is a cheaper and a little bit less accurate, but also high resolution mass spectrometry. And again, we are very far from biology here. We are more in the physics, so we are interested in reading the contents of a full transient. Well, this means basically that we have signals from all the ions present in the sample, and we need to decipher, to decompose these signals by Fourier transform operation. So mass spectrometry information gives us the empirical mass of any metal ligand compound in a biological system. So here it's important to understand the concept of mass accuracy. The more accurate is our measurement, the fewer possible empiric formula we may have. That's why not only resolution is important, but also accuracy. And as you can see, if we've got a species of 400 mass, then if we measure 1 ppm, we've got a single molecular mass, empirical molecular mass. So that's called data analysis. We search for metal-specific isotope patterns, mass defects, to get out of this set of 10 million experimental points, 500 or maybe 1,000 that are of interest of us. And then if we eliminate all the artifacts out of this 1,000, we may get maybe 100 of meaningful metal complexes, and then we identify them by this tandem or particular transformants that grown in the presence of large concentrations of nickel. We carried out the same speciation, but it was not on the plant like here, but it was on yeast, and we found exactly identical mass spectrum, which meant that this nicosyanamine ligand is functional. And once you've got yeast, it's very easy. You can sequence this yeast, and then you can see that there is one particular gene that is the gene coding for nicotinamide synthase in this case, because it came from the library of the plant that was expressed. So as you know, to bacterial infection, and this response is called nutritional immunity. So what is the purpose of this nutritional immunity? Is to render metal ions inaccessible to the pathogen, and they can be rendered inaccessible by using strong chelators. So these chelators must have to be stronger than the mechanism bacteria had developed to acquire the trace elements, such as, for instance, iron from the environment. So this the pathogen invented these mechanisms, pathogenic invader. So the example will concern here Staphylococcus aureus. It's a gram-positive bacterium associated with respiratory infection, associated with food poisoning. And we will answer the two questions that I had announced. I said, of data treatment. And you see what you can see here. Then this bacteria produced a ligand. This ligand is common downstream of this nicotinamide synthase gene. So then if we remove this gene related to this nicotinamide synthesis, what we observed that there was no synthesis of staphylopin. So it means that the hypothesis came true, and this means that this is the right place to look in the genome. So then looking upstream, downstream, then you've got well, 3 genes. You can construct an operon that will contain one gene downstream, one upstream. And then when this operon is expressed in Escherichia coli, suddenly we observed a beautiful signal of the nickel complex with staphylopin. And here we come to the mechanisms, to the enzymes of 3 enzymes, so each coded by these different genes. And these enzymes, when they are applied sequentially, that first one, it converts L-histidine to D-histidine. Then the second one adds an aminobutyric acid moiety, so this nicotinamide moiety. The third one, it's reductive condensation of the product with alpha keto acid, and then it produces staphylopin. So that's really a beautiful example of the synergy of the two conceptual approaches that make the essence of metallomics. The first is advanced analytical chemistry. Advanced analytical chemistry, it means a map of all the metal species present in a sample, and this can be obtained by the coupling of multidimensional separation techniques with bidimensional mass spectrometry. When I say bidimensional, on one hand it's element-specific ICP-MS, and on the other hand is molecule-specific electrospray with multi-stage fragmentation for structure identification. So that's the analytical part, and then the holistic approach, assembling parts to functional units, so how metal ions work in the system. And here it's a hypothesis-driven approach. So you need many hypotheses to be put forward. You need collaborations, good collaborations with geneticists, with biochemists, with plant physiologists. So it's really the teams, well, are 15, 20 people teams. And this is not the purpose, you know, of this lecture to discuss the details. I will just wanted to underline this. as this dual metallomics concept so beautifully expressed in the primer to metallomics published by Professor Wolfgang Maret a couple of years ago. So here is the people who contributed to this study. So in terms of co-authors, I used people I work with in my place, at my institute. That is a beautiful building that you see here in the picture. We are 300 people in the institute with all the different disciplines, so chemistry, theoretical, analytical, physical, theoretical chemistry, polymer chemistry, and microbiology. But for this particular study, we searched for collaborations in different places in France, because in France we've got the possibility to get a big national grant for multi-team, multidisciplinary research. That was the case here. So I acknowledge people from Marseille, Montpellier, Paris, and Aix-Marseille. And I would like to thank you all for your attention. Thank you very much.
Thank you, Professor Lewinsky. You have completed your lectures within the time limit. Normally, we don't put questions to the keynote speaker, but it seems our audience are very, very attentive and very curious to put some questions. So 2 or 3 lead questions may be asked if you have any inquiry to Professor Lewinsky, please.
Yeah, a good talk, Professor Lewinsky, for eye-opening, and it looks like you are throwing more of an opportunities to us or to the world of science. And one thing that I want to know in the beginning is that how do you define a metallome? You have talked about metallomics. Then my further questions will follow after that. Yeah. What is a metallome?
Okay. So for me, metallome, well, the term metallome was first coined by the late Robert Williams, who was the founder of bioinorganic chemistry. And it was defined as the inorganic complement of genes in a living cell. So that was the basic definition. This definition has evolved a little bit, but the essence is the same. So it's a global view of, well, it's a set of concentrations of metal ions and metal species in a biological system. So for me, this is a metallome, and so this is complementary inorganic component to the genome. So for me, this definition is summarized. I think that I made things completely wrong, but if I come to the slide,
there was a definition. Okay, several.
Yeah. So this is the definition, basically. Yeah. So the definition you can find in this International Pure Chemistry paper, yeah. But the essence is here. So you've got metal ions.
Yeah, there. It should have been there.
Embedded in a genome, proteome, metallome. All these interactions.
Okay. Yeah. Now, yeah, understood. The question arises because I work in the plant model for various things and other things. Now, there is a synergy here. Because you are coming from a center, from a country where there are less bioresources. Now we are in a system where there is a huge bioresource. Now, in terms of nutraceuticals derived from plants or from microbes, we concentrate on certain individual plants of high value. Now there lies a problem of identifying or creation of new species or new types. Let us call it genotypes, chemotype, etc., etc. So in this kind of breeding technique, we use certain techniques called as tilling. So we use mutants or mutagens to develop mutants in a very, very specific region of the genome to create diversity or variations. So if your metallomics driven creation of certain genotypes or different types, then there's a synergy which can bring about lots of, we can pinpoint it, let it be, a plant having a high concentration of a very, very medicinal important plant, then this particular approach in a reverse way can have lots of synergy. That is what.
Yeah, well, I think that the key information here is that you are able to correlate a particular set of metal ion concentrations with a particular genome. Yes. Once you manage to do it, well, you can do it in the lab very easily because, well, you've got the collection of mutants and then you can correlate them. In nature, well, there are many, and then you control the system because you know what you put in hydroponic. In real environment, you do not control. So you can just, well, pick up plants that you find particularly interested in getting a particular element from the environment. So one of such cases is the case of hyperaccumulation. You've got some plants that like metals, but hyperaccumulation is more about toxicity, and here we are talking about essentiality. So if you could imagine correlating a plant specimen with a particular grid for a particular ion, that I think it's extremely important. So this is what was done for this bacteria. If you can do similar things, that if there are some particular plants with some particular genes that privilege the acquisition of a specific essential element from the environment, that would be really a great conclusion.
Only one question, please, from the audience.
Thank you so much, Professor Lominsky, for such an elaborate session. Really, I just come to know about genomics, transcriptomics, proteomics, metabolomics, and all the omics that you have mentioned regarding metallomics, ionomics, metalloproteomics also. These are all new things to me. And one question regarding the speciation, which is very much interested to me, is regarding one example that you have cited, ZNS, and which is producing the enzyme nicotinamine synthase, and it is going to bind with the ligand nicotinamidase, and which is nickel resistance. So in the study that you have shown, sir, my question is, so which ligand specifically bind to which metal ion? So what type of analytical approach that you apply? And instead of this nickel resistance, why not you study for iron or copper or zinc?
Yeah, well, nicotinamide, about this time, I don't remember the exact dates, but it must have been the same year or a year before. There was a study in Japan that found nicotinamide as a transport ion for essential elements, especially for nickel. So the ligand is nicotinamide. Yeah, this is the ligand. This is nicotinamide. So now, 20 years later, you will find in the literature plenty of papers demonstrating the importance of this ligand in transport of metal elements. We did even one, I think, I skipped this slide because I didn't have time. That's no, I don't have this slide. But no, this is not nicotinamide, so I don't have this slide here. But it was about essentiality. And that made us to study the role. Well, actually, we didn't know that it would be nicotinamide. We studied nickel resistance of class picarulescence. But we found this nicotinamide as the main nickel complex. And then we got interested in the genetic background of the synthesis of this nicotinamide. The ligand is nicotinamide. The complex is 1:1, zinc to nicotinamide. There are many varieties now of nicotinamide. So there are hydroxy, dihydroxy, many different ones. I can send you more detailed information. We studied a lot of nicotinamide varieties, in Peru, in areas with a lot of hyperaccumulating plants, and we found that nicotinamide was really a very interesting ligand. So to answer your question, the ligand is nicotinamide. We talk about 1:1 complexes. The analytical approach is mass spectrometry, the one that we have developed.
Well, there may be many more questions, but I don't allow them
thank
because
you.
we are already
Thank you,
in
sir.
the time schedule. Thank you, Professor Levin.
Sorry, one more thing. I saw
Yes.
people were taking pictures. Well, if you send me a mail, then I will send you the presentation. The quality will be better.
Right. Thank you, Professor Levinsky. You have really unraveled the mechanism of molecular mechanism, metal uptake and transport in plants and bacterial systems. And the study seems to be a very complex one. It involves a very advanced analytical chemistry, physics, mathematics, and statistics and all sorts of things. And we are really, our students, audience, I'm pretty sure you are enriched with the many sophisticated equipments needed for this study. And this is probably the beginning for our scholars. They will pursue the matter in collaborations with Professor Levinsky in future. Thank you, Professor Levinsky, once again. Thank you very much.
Thank you, Professor. Thank you, Professor Levinsky, for the lecture. I'm sure your lecture has been remarkably insightful for all the participants attending the conference. May I kindly request Professor Shetty to kindly felicitate Professor Levinsky. Professor Levinsky, we would like to felicitate you. A big round of applause, ladies and gentlemen.
I will send you the presentations, you send me the picture. Thank you very much. Thank you.
I would also like to thank Professor Shetty. May I now invite Professor T.H. Binoy Singh to kindly felicitate the chair, Professor Shetty. Thank you, sir. Now we shall commence our technical session 1 on the theme Omics in Metalomics and Allied Sciences. This is a parallel session, so there will be oral presentation. Oral presentation OP1 to OP4 will be here, and parallel session, the oral presentation will be from OP5 to OP11. So those presenters who are on the other session, that is in the conference hall in Department of Life Science Zoology, may I request you to submit the presentation to Dr. Santa. He will be responsible for that. So in technical session 1, we will have a plenary lecture by Professor N. Saha, Dean, School of Life Sciences, Northeast Hill University, Shillong. It will be chaired by Professor Sagartirtha Sarkar and co-chaired by Dr. Amit Seth, Department of Life Sciences, Botany, Manipur University. The presenters on the other venue, may I kindly request you to go to conference hall in Department of Life Sciences Zoology. Now I hand over the session to the chair and co-chair.
this one? Yeah.
Good afternoon. So, in the technical session 1, we have a first plenary lecture by Professor Nirmalendu Saha from Department of Zoology, Northeast Hill University, Shillong. And Professor Saha is now presently the Dean of the School of Life Sciences and Professor of Zoology, Nehu. And he has almost 34 years of teaching experiences in the field of biochemical adaptation, biological chemistry, animal physiology, molecular biology at postgraduate level. His field of specialization is biochemical adaptation and metabolic regulation. So he is a recipient of the Fellow of National Academy of Sciences, Allahabad, Fellow of Zoological Society, INSA Young Scientist Award by Indian Science Congress, DAD Fellowship for Postdoctoral Research, and Senior DAD Fellowship twice during 1997 and 2006. Published more than 100 research papers in journals of repute, guided 22 PhD scholars so far. He has successfully completed 12 research projects as a principal investigator and presented talks in different esteemed conferences. So with these words, I would like Professor Saha to start his deliberation. Thank you.
Thank you very much, Professor Sarkar, for introducing me. Before I start, I don't want to do too much of formalities. Already we're running shortage of time. First of all, I would like to congratulate, thanks the organizing committee to invite me and giving a lecture and share some of my research work which I carry out in Northeastern Hill University. So you see, in earlier lecture, in the keynote address, you have listened a very excellent lecture by Levensky. He mentioned about that trace element in the biological system, how it is to be analyzed, identify, and quantify. We are a pure biologist. We do not understand about technical part of it, how to trace all these things. We use all these technology and we apply in our biological system. We try to understand what is the physiological biochemical aspect of these molecules inside the biological system. And I work with a fish as a model. So I'll take fish as a model to explain that some of the metal oxide nanoparticles, because it suits with the metallomics, that's why I decided to talk on this, because I do lots of work with nanoparticles and see their effects on the biological system and what are the toxic effects it causes. And I'll mainly concentrating on only one aspect, that is metal nanoparticles causes oxidative stress, and what is the adaptive mechanisms we find in the biological system to tackle this problem and how they survive up to a certain extent. But if you go beyond certain limit, nobody can survive, they'll die. Up to the tolerance limit, because all living organisms has got their own defensive mechanisms. Okay, for all these things, you see, virus and all, first our immune system goes activated and we kill it. If it doesn't work, then we go for external sources how to kill it. So here also, we'll talk about it, stress that is being also mentioned in that introduction section. You see, we have been using these metals like copper and all from time immemorial for various purposes, mainly for the human welfare. But we never think about what are the hazards it will cause if we use in excess. Likewise, nowadays it has come, these nanoparticles are being used for various purposes, not only metal oxides. See, for the benefit of the human welfare, is not it? Various types of cosmetics, ladies uses. They use lots of nanoparticles are used for synthesis of these all cosmetic materials. Pharmaceutical products, they are being used regularly and in a huge amount. But we forget about what are the hazards it is going to be caused into the environment as well as the human welfare. This is a dangerous thing. We think later when it crosses beyond certain limit, like plastic, polythene. We are very happy with the polythene, you see, and started using for every day-to-day purpose polythene. Now we know the consequences now. We are really fed up with it, how to take care of it, and we are finding some. But that's why when you are using or something, first thing one should think about the waste product, whether it is causing some harmful effects to the human welfare as well as other living organisms, because you are disturbing it. So human being has to survive, other organism also has to survive. Otherwise there will be disturbances of the nature. Okay, so I'll not go into that. I'll talk only about these certain nanoparticles, that is only two, zinc oxide nanoparticles and titanium dioxide nanoparticles, whether it causes oxidative stress or not, and how that we can take care of with this oxidative stress, what is the defensive mechanism we have in our body. You see, just why I just told nanoparticles, of nano is a Greek word, which means extremely small particles, and the size varies from 1 to 100 nanometer. Those molecules, we call it nanoparticles. If you just look at the structure, size of the water, it is 0.1 nanometer. Glucose, 1 nanometer. Whereas, if you just see nanoparticles and viruses about 100 nanometer, within these 1 to 100 nanometer size, what are the things that comes, we call it nanoparticles, and you can see the sizes of different molecules. You see, these are the various sources of nanoparticles. You can see we are using it for multiple purposes, and you get so much of nanoparticles for our human benefit, for our welfare, but at the same time, lots of waste products are coming into the environment and which are getting accumulated not only into the water, also into the environment, and it is getting accumulated where system, food chain, and we know the consequence of it. It can cause various effects, because you see, if you look at this bulk size, because zinc, there's a bulk, there's a zinc oxide, nano, you can convert it to nano. Okay, there is a techniques to convert it, nanoparticles. If you compare it, these nanoparticles are highly reactive compared to the bulk material. Chemists can tell better about me, than me why it is highly reactive. Okay, so I've just summarized some of these, whatever, large surface area per unit mass, potential high surface reactivity, hardness, electric conductivity properties, magnetic properties, electron band gap, random molecular motion. So due to all these reasons, they are more reactive than the bulk material. Okay, so that is why they are more reactive. And then I'll tell you in brief how this nano material or metal oxide nanoparticles can enter into the cellular system, mostly by endocytosis. So here, by phagocytosis it can enter, then clathrin-mediated endocytosis it can enter, by micropinocytosis it can enter, by pinocytosis it can enter. It forms a vesicles, causes various damages in your body, then they go out. Okay, by some means, by exocytosis. Okay, the damage is caused, then only they go out. Is not it? So that's why you see we are using it, but we never think about what are the consequences we have into the biological or cellular system. Now, if we just see that, you see, these are how these all reactive oxygen radicals are being formed. These are some of the examples of reactive oxygen species. This is singlet oxygen, superoxide anion radicals, hydroxyl radical, alkoxyl radicals, peroxyl radicals, hydrogen peroxide, lipid peroxide. See, this, if you just look at it, these free radicals, 1 electrons are missing in the outer shell. That is why it is so reactive. Once you use some antioxidant substances, you see, these electrons are donated here and becomes stable, the action is less. Okay, when it is unstable, so it causes more damage to the biological system. So now, in the biological system, how this ROS could be produced by some external sources, by pollutants or nanoparticles, whatever it comes. So through electron transport chain, oxygenase, auto-oxidation, specific system, NADPase oxidase system, exposure to pollutants, all these leads to ROS, reactive oxygen species formation. I will not say always ROS is bad. ROS, it has got some function. It also can act as a signaling molecule inside the body. They stimulate various beneficial processes, metabolic processes. But if it produces in excess, that is dangerous. That causes oxidative stress to your cellular system. Okay, now this, if it is produced in excess, this can be regulated by your system inside the body by some low molecular mass scavengers, and also high molecular mass antioxidant. There we have various enzyme system in our body. These are large molecular mass body. They can take care of these excess of ROS, reactive oxygen species. So this is what are the consequences. See, this causes damages to the cellular constituents, causes cellular injury, apoptosis, and necrosis. At the same time, you have a regulatory system. You have a mechanism to regulate it. Regulation system gets activated. Adaptation to internal and environmental changes takes place to a certain extent, to a certain limit. If it crosses the limit, then you know the consequences. Then, not only that, this oxygen radicals also has some beneficial effects. It can attack all these external organisms, some infectious bacteria, viruses enters, they can kill those organisms to a certain extent. So that's why I say it is not only harmful, it has also got some beneficial effects to the cellular system. You see, ROS is produced, but when it is produced in excess, that is gets unbalanced. All your system homeostasis gets unbalanced. Redox homeostasis gets unbalanced. Now how to make it balanced? There are various ways to balance it. First, see, these are the small molecules, micromolecules, hydroxycinamides, glutathione, bioflavonoids participates in these to neutralizing. They give one electron and then that gets stabilized. Then carotenoids, vitamins also. There are types of vitamins, vitamin C and all, they can neutralize to some extent all these oxygen radicals which are being formed inside the body due to some reason or the other. Then these macromolecules, that is antioxidants and all, they get activated, they try to neutralize to some extent. The next one, nitric oxide, this is our hypothesis. You know that nitric oxide is a signaling molecule. They play a very vital role in adaptation to various stresses, not only in higher vertebrates, also in lower vertebrates. I've been working with nitric oxide molecules for quite some time, and we have published many papers related to this, how it helps in adaptation to various stresses. So we thought that nitric oxide is also playing a very important role in neutralizing these all oxygen radicals inside the cellular system. I'll explain with experimental evidence that it really plays an important role. Okay, then this redox homeostasis can be neutralized by all these methods and cell is safe. Okay, cell is normal. Now, I work with fish 35 years with this. I've done lots of work with nitrogen metabolism with this fish, and I've shown for the first time the presence of a functional ornithinuria cycle in this fish. No one reported it. Earlier, we know that there is no functional ornithinuria cycle in fish. We have shown it. Not only that, it can be upregulated also under ammonia stress and various other stresses. Then I took the same model to explain what are the antioxidant mechanisms that are present in this fish to tackle the metal oxide induced oxidative stress. Why these fish are unique? These air-breather magur fish are facultative air breathers that are found predominantly in the tropical Southeast Asia, predominantly found, published already some of it. This is this keeping all these objective. Whether the exposure of metal oxide nanoparticle, that is 2, we have been working on other nanoparticles also, but I'll explain with zinc oxide and titanium dioxide nanoparticles causes any oxidative stress or not. First, you have to see whether really it is causing oxidative stress or not. Then, what are the possible antioxidant stresses that are being 96-hour LC50 value? We have to see when you treat it, you should not give too much of concentration that the fish die or the organism die. Then your experiment is failure. Is not it? So we have given 10 times, 5 times lesser than that, so that it is tolerable limit. And then we analyze various parameters, what I mentioned, whether it is causing oxidative stress, what are the antioxidant strategy, how it gets activated, what is the role of nitric oxide in detoxification of these reactive oxygen species, and also HSP70, whether it is getting induced under these conditions, okay, to adapt in a better way. So before we go to that, we purchase these nanoparticles from Sigma, okay, because that's commercial source. We wanted to know whether they are really nanoparticles or not. Through electron transmitted electron microscope, we have measured The average diameter is about 45 nanometer, this one, zinc oxide, and titanium dioxide is 41.5 nanometer. That means they are nanoparticles. Okay, then I'll not go detail much about it. Then what we did, we measured different parameters which gives an idea about whether it is causing an oxidative stress or not when you are treating with zinc oxide. You see, we measured in different tissues, okay? H2O2 is one of the measure parameters. If it goes up, we can call that oxidative stress has taken place. MDM, malondihyde, dehydrogenase, that is comes from the lipid peroxidation. If it goes up, that means some oxidative stress has taken place due to generation of ROS. Even glutathione also is another parameter. So we measured all these parameters. We found that treatment of zinc oxide causes significant rise of all these parameters, initially followed by gradual decrease of all these parameters. That means it is slowly getting adapted. So it is bringing down all these reactive oxygen species so that they can survive under this stressful condition. You can see it. It has gone up. You see, this is the one you see. It has gone up, then it is slowly going down. Okay, so then 3 days, 7 days, and 14 days we have seen it. So likewise, same thing we got it for MDA as well as GSS. Now, we also measure certain macromolecules, antioxidant macromolecules, whether these activities are going up or not under this stressful condition, that is zinc oxide, nanoparticles. Interesting, you can see in most of the tissues, this got activated. Initially, there was a decrease, you see here, 3 days, it is a decrease followed by gradual increase in most of the tissues. Likewise, catalase also. You know, SOD and catalase is the first line of defense against the oxidative stress. Then comes other glutathione related pathways comes, okay. Then, so those things comes one after another, but SOD and catalase is the first line of defense against the oxidative stress. So we measured this one as well as glutathione related enzymes. So we found that this enzyme's activity is getting upregulated under zinc oxide nanoparticle stress. Then also we measured whether these changes is due to activation of specific genes for all these enzymes or not. We measured the mRNA expression of all these genes. This is SOD1, SOD2, catalase, GST, glutathione peroxidase, then thyrodoxine reductase. So we found out that these are also getting upregulated. And I would like to mention here another very important finding we have just reported. This is thyrodoxine reductase. Normally, in our body, we have glutathione reductase. It's one of the pathway for glutathione synthesis, but this fish has got thyrodoxine reductase in place of glutathione reductase. This we have studied thoroughly, we have sequenced it, we have cloned it, we synthesized the protein, we have done all biochemical characteristics and kinetics, we have done it, and recently we have published in International Journal of Biological Macromolecules. So they have accepted that we do not have glutathione reductase, but they have glutathione thyroreductase, okay? And probably it has got some evolutionary significance, and this gene normally found in lower vertebrates, not in vertebrates. Also, we wanted to know that these transcription products are getting really translated to proteins or not. We measured different proteins, enzymes, that also got upregulated. I will not go into detail. You can see band intensity has gone up at time intervals. So that has caused the upregulation of different translational product of all these genes. Then what we did, now come to the nitric oxide. We wanted to see first whether this zinc oxide nanoparticle is really inducing the production of nitric oxide or not. So we measured it, nitric oxide level in different tissues, and this inducible nitric oxide synthase is one of the enzymes. There are 3 isoforms, neuronal, endothelial, and inducible nitric oxide synthase, which causes the production of nitric oxide. It is also getting upregulated under zinc oxide treatment. So then also we studied this gene expression study of this iones. It is also getting upregulated. You can see even the translation of product. See, initially no enos, we cannot find enos activity in control fish. But once you treat it, it appears. You see, it has started appearing, and at later stages more expression of iones activity. So that means it is due to induction of iones the NO is getting, synthesis is getting stimulated and it is getting accumulated into. But it doesn't say what it plays a role in antioxidant activity. I'll come to that for later set of experiment with isolated hepatocytes. We have done it. We have proved that NO plays a very important role for antioxidant activity. Okay. Now, again, as I mentioned, we have also measured different SSP gene, heat shock protein genes. We took SSP70 and SSP90. Two isoforms are there. This is inducible one, this is constitutive one. This is inducible and this is constitutive one. We found that most of it got upregulated under zinc oxide nanoparticle treatment. And we have seen the protein expression also has gone up. Okay, so that is causing this induction of some important heat shock proteins. So probably they are helping in renaturation of some denatured protein that has caused due to accumulation of reactive oxygen species inside the cellular system. Now, how to prove that nitric oxide plays an important role for antioxidant activity? We took the in vitro experiment approach. We isolate the hepatocytes. We did primary hepatocyte culture, and then because we had to use certain inhibitors. Otherwise, you cannot definitely say that these things are involved in that. So it is not possible in whole animal to use certain inhibitors. So we have done it in the liver because that is the most metabolically active tissue. See what we did it. We isolated the cells and treated this cell separately with zinc oxide and titanium dioxide nanoparticles alone. Then we took certain inhibitors. Aminoguanidine, which is a specific inhibitor for INOS, inducible nitric oxide synthesis, we blocked the INOS so that NO synthesis goes down. Then also we used another inhibitor, that is the BAY, which is a BAY element, which is the inhibitor for NF-kappa B, that is a transcription factor. And we know very well that NF-kappa B is the major transcription factor which induces the INS activity. That is, we have shown it, okay? So that's why we blocked one way with this enzyme itself, then we blocked NF-kappa B so that it cannot induce this INS enzyme expression. So, and also another side we use inhibitor for MAP kinase signaling pathways, different isoform, different inhibitors we have used it, and different parameters we measured after treating these at different time intervals to prove that NO plays an important role in antioxidant activity. These are some of the data which you have got from this experiment. We did two sets of zinc oxide nanoparticles, 5 as well as 10 microgram per ml. Okay, in the hepatocyte, you can see this one, zinc oxide, this is the green one, and then at 10 micromolar, it is a purple color. You see here, 10 micromolar is green, and this one is white. You see here, it is going up initially. This H2O2 level is going up, and then it is going down. Likewise, at 10 micromolar concentration, also same thing is happening. The green one is going up, then it is going down. But when you use these two inhibitors, instead of going down, it has further increased this H2O2 level. It's not it. That means it is not. So once you block NO production, there is a further induction of oxygen radical formation. That means your adaptive system is these two inhibitors, whether it has got effect of these antioxidant enzymes or not. That is the first line difference, SOD and catalase. Once we blocked it, so see, only zinc oxide causes induction, as we have seen in the whole animal. Here also in the primary hepatocyte, it went up, but when you use these inhibitors, see, instead of increasing, it is going down. There's a down regulation of these two enzymes, enzyme activity as well as your transcriptional activities also, mRNA level also is going down. You can see here in both SOD, there are two isoforms, SOD1, SOD2 and catalase. See, it is going down when you block the synthesis of NO. And so the translational product of these two SOD-NO, same story here. Zinc oxide 5 and 10 microgram per ml, see there is upregulation. You see it has gone up, both the. But once you block, when you add aminoguanidine as well as BA, see there is a downregulation. Okay, this enzyme synthesis also is going down if you block the synthesis of NO.
Dr. Shah,
Yeah.
kindly wrap up within 5 minutes, please.
Yeah, it will take some time. Okay, just 1 minute. Okay, so I have some more data. Yeah, you see, this also, we have done immunocytochemical analysis. Okay, you have used immunostained, and we have stained it. You can see this is with zinc oxide, but once you use these inhibitors, you see there is no, see, you cannot see SOD much. The red one is SOD, blue one is the nucleus. See again 10, so much of, see, this SOD protein, but once you have used this, it has gone down. Likewise, catalase also same thing. See, this is gone up, but in presence of inhibitor, it has gone down. So now we come to this formation of NO under these conditions in presence of inhibitors and all. We have seen it with zinc oxide causes increase of NO synthesis, but once you use these inhibitors, so there's not much of increase of NO production. Okay, then we have also measured ionase activity under this condition, experimental conditions. You can see zinc oxide is getting increase of ionase activity. Red one is the ionase protein. Whereas if we just use BA, there's not much of increase, okay, both in the 5 and 10 micromolar zinc oxide. Now, this NF kappa B also we have seen the expression. This is a transcription factor. What happens? It has gone up. You see, P65 has gone up. This is one of the subunit of NF kappa B. And then you see protein expression also. And you see that if you look at the translocation, so this is the control. Normally it is not present, very less amount you put. One, you have to induce it. Once you have induced, you see so much as formation. Red one is the NF kappa P65. And then after that, it start getting translating into the nucleus. It is going in because it has to hit the specific gene, then only it gets activated. So it goes into that, into the nucleus, and slowly disappears. Same thing, okay? At later stages, if you see, slowly it is disappearing. That means it appears and disappears. Appears, activate the gene, and disappears. This is what has happened under this zinc oxide treatment, nanoparticle treatment. So, again, I'll tell in brief, we have done the experiment with titanium dioxide nanoparticles also. We have got the similar results. Similar, I'll not go into detail of all these. We have done in detail about all these things. We found same thing, titanium dioxide causes more of oxidative stress. At the same time, antioxidant gene goes up. If you block it, NO production, more of oxidative stress. Then this NO production also, we have seen it under titanium dioxide. Story is almost same. I will not go into detail due to time constraint. See, it is going up, and then when you use the inhibitor, the NO synthesis is blocked. Then another pathway, MAP kinase signaling pathway, that is your ERK, P38, and Jun kinase, JNK. We have seen it. If you use these specific inhibitors, when you treat with titanium dioxide, it is going up, but when you use specific inhibitors, that inhibitor for P38, inhibitor for ERK and JNK, see there is a down regulation. All these signaling molecules are going down regulated. So same thing we have shown by immunostaining, we have shown the same thing, definitely that this protein expression is going down under these treated conditions. Now, I would like to explain with this model what we want to propose.
Sir, your time is up.
Let me explain this one then. Okay, this is a invited lecture. I think you have to a little bit, yeah? So let me finish it.
Then I have no problem if the organizers are ready.
I have to finish it. Otherwise, it is half a story. Okay, see, if you just see zinc oxide and titanium dioxide, if you take it, it goes into the cell, it causes reactive oxygen species formation. Is not it? Then it causes induction of NF kappa B, and NF kappa is going down, hits the specific gene, gets upregulated the enos. Enos is getting NOS2 mRNA expression, more of ionase, more of nitric oxide synthesis. That is causing induction of SOD and catalase activity, and this RO is getting converted to H2O2, and ultimately water that is getting neutralized. Another is that NO, probably NF kappa B is also activating this glutamate to reduce oxidative stress. Thank you very much. So this is my some of the co-workers, I just want to say, in my lab, they are working. Thank you.
A very nice presentation, sir. Yeah, I just want to ask some questions related to the oxidative stress. You mentioned that the exposure of the zinc oxide or the titanium oxide nanoparticles decrease in oxygen system, and after some time it also increase. And you talk about
what you said. I just goes down regulated because they cause some damages. You see, because probably these are also protein.
Okay.
This initial system fails because suddenly they get a shock. ROS goes up. That causes denaturation of some of the proteins, including these enzymes also. Okay, that is why it goes down. Then followed by, due to some way or the other, as I told you, nitrogen of all these genes.
Sir, what I'm thinking is like, suppose if you keep for the nanoparticles for a long period of time, after increase of your thioredoxin reductase, if you keep for nanoparticles for a longer period of time, it may also decrease. That leads
limit for everything, you see.
Okay. Yeah, thank you, sir.
The chair to kindly felicitate the lecturer.
Yeah.
Kindly.
Thank you. Thank you very much. Thank you. Thank you.
Thank you, sir. Now we shall start.
Pointer, yeah. Namaskar. Respected Chairperson, Professor Sagartitha Sarkar sir, Co-Chair, Dr. Amit Seth sir, honorable dignitaries and research scholars and guests present over here. I am Prajalta Patir, and today the topic of my presentation would be prevalence of hypertension, it's hypertension. If I were to talk about previous works which have been done for our country, this is just a glimpse of the works that have been done by various authors and the prevalence which they have reported. If I were to talk about the recent status of our country, then these are few of the recent papers which I could find. A study done by Ramakrishna et al. 2019, and basically their choice of study, their area of study was Kerala, and they have reported a prevalence of 11.2%. The thing to be noted here in their study was that the age group which they have chosen for their study was the young adult category, that is from 17 to below 40 years of age. So in that particular age group, the prevalence of hypertension that is being reported as 11.2 is quite on the high side. Another study by Abriga at the entire northeastern states of India are facing a high prevalence of hypertension. Now here, the number 6 one, that is Mishra et al. 2014, they are the only one paper which I could find throughout the literature who have done on the missing community, the tribe that I am working on. And actually, their study was based on the risk profiling of NCDs rather than hypertension, but still they have reported a prevalence of hypertension to be 26% in the missing community of Tinsukia district. So the place where I've worked, that comes under Dhemaji district of the state of Assam, India. So the main objective of taking up this study was to study the prevalence of hypertension among the missing tribe of Gogamukh region, Assam, and this comes under Dhemaji district of Assam, to uncover the risk factors leading to hypertension among this study group. Study area comprised of villages selected randomly under Naharbari Gram Panchayat, Gogamukh, and most of the sub-study subjects were from Tingiri village, and some were also from the Parghat village. Both of them, they come under the Naharbari Gram Panchayat. Sampling and data collection was done between the period of December 2018 and February 2019, then from August 2019 and October 2019. Random sampling of the subjects was done, and a total of 384 subjects were chosen at random for this study. Now here the study time period is December 2018 to 2019, because my ethical, I have obtained the ethical clearance from Institutional Ethical Committee, Sikkim University, and this project actually is an extension of my dissertation work, which I was doing back in that year. So there are some parameters on which I have prepared the questionnaire and collected data from the subjects. Those were age, gender, BMI, smoking, alcohol intake, tobacco chewing, then extra dietary salt intake, diet, what kind of a diet they prefer. That is, they have more vegetables and fruits in their diet, or more meats and fish in their diet, or oily foods in their diet. Then physical activity, what sort of a physical activity? Is it on the moderate side, or is it on the strenuous side, or is it on the sedentary side, was also collected from the willing participants. Awareness about their condition and what could be the consequences of hypertension was also collected from the participants. Then medication, whether the hypertensives found were on some kind of a medication or not, and whether their BP is under control. This is assessment of BMI. Measurement of blood pressure was taken by mercury sphygmomanometer, and I had the help of the one of the health officials from the Naharbai Gram Panchayat, PHC.
Ma'am, you have to wrap up. Time is almost up.
Okay. So the prevalence of hypertension was, sir, just 5 minutes, I'll wrap up. Just
Not even 5 minutes.
okay, sir. So prevalence of hypertension was found to be 20.3%, that is 78 out of the total 384 subjects were hypertensives. Then prevalence was found to be significantly associated with age, BMI, alcohol intake, serial smoking, extra intake of dietary salt, and less physical activity. Diet consisting of red meat, fat-rich and oily food possesses a high risk to hypertension. Actually, it is being reported by many authors, but in my study, I did not find a significant association of diet to the prevalence of hypertension. Family history was also found among the subjects on the case of hypertension, and awareness on the perils of hypertension was high, but people had a very laid-back attitude to the condition. So this is one ethnomedicinal control which is being practiced by the missing community back in Assam. That is, they will take the leaves of Clerodendron species, which is known as nefafu in Assamese and pakkom in the missing dialect, and they will just stew it along with garlic. They make kind of a stew which they will consume, which they will take with rice, and it is believed that it reduces blood pressure, which to some extent is believable because many works are being done for Clerodendron species, and authors have reported that it does actually have antihypertensive property. Hypertension is known as a risk factor to diseases and conditions like diabetes, heart attack, stroke, paralytic attack, and paralysis. Though hypertension is not the sole reason for these diseases, but it is one of the major contributing factors for it. Prevalence of hypertension has reached far-flung areas of the country, and the prevalence has been rising ever since. And so there, along with awareness, along with BP control, awareness on the condition, on the perils of it is highly the need of the hour for these days.
Ma'am, thank you. To wrap up, please.
So with this, I would like to wrap up my presentation. Thank you so much.
Thank you. May I have the second presenter ready, please, for presentation OP2? Kindly come on to the stage.
A very good afternoon to the chairpersons and all the attendees present in this hall today. I'm Dr. Reena Haubram from the Department of Biotechnology, Manipur University. The topic that I'm presenting today is the study of the neuroprotective property of some of the medicinal plants in Manipur. These are traditionally used medicinal plants for improving neuromuscular function, which are used in by the traditional healers in Manipur. The first one is Albizia sinensis, the second one is Plumeria rubra, and the third one is Silex tetraspermia. So these are the methods that I have used. I have prepared the methanol crude extract for these plant samples. Then initially we do the antioxidant studies using DPPH and ABTS assay. And for the neuroprotective study, we use a cell line, which is the SHSY5Y cell line, which we. So these are my results. So in the initial antioxidant studies using ABTS and DPPH assay, all the 3 extracts are shown to show good good antioxidant activity. So all the 3, we compare it with ascorbic acid as a standard, and we have seen that in the first in the first 3 graphs, we have seen the antioxidant activity compared with the ascorbic acid. We can see the increase in the antioxidant activity in both the extract, except for this. This has a little bit lower kind of antioxidant activity. Similarly, in the PPH assay also, we have seen comparative amount of antioxidant activity of the 3 extracts. Then this is a figure representing the differentiation of the neuronal cell line. This is SHSY5Y cell, which is differentiated with the expression of neurites. And then we do the MTT assay. We have different groups, control groups. Then the rotenone is a toxin that we use to establish a neurotoxicity. And then we have the different doses of the extracts. So different doses have shown to give protective increase in cell viability after the after treatment. So during the treatment program, we usually give a pre-treatment of the extract 1 hour prior to rotenone treatment, and we observe the viability after 24 hours. So this supports the neuronal survival. This is the control groups where MAP2 is significantly expressed. And then in the rotenone treatment group, we can see decrease in the expression of the MAP2. And then in the 3 groups where we have used the plant extract, we have shown the increased expression of the MAP2. And we have quantified by counting the cells in different groups of these cells. We have repeated this experiment at least 3 times and taken different areas in the plates to count the cell number of cells expressing MAP2 and quantitatively express it. This shows the intracellular ROS level. In the control group, it is low. This is the DCFDA assay, where DCFDA inflorescence will be increased when there is oxidative stress. So it represents the ROS expression. So compared to the control, the rotenone group has high expression of the fluorescence, DCF fluorescence, but after treatment with the extracts, we can observe that decrease in the fluorescent intensity is there. So we have quantified this by using ImageJ software, and see the fluorescent intensity being decreased after the treatment of the extracts in all the 3 groups. The next is for the mitochondrial membrane potential. We use the rhodamine 1,2,3 dye. In control, when there is a stable mitochondrial membrane potential, the expression of the rhodamine 1,2,3 will be increased. But when the reduction is there in the potential, there will be decrease in intensity of the fluorescence, which is rectified after the treatment of the drugs or the extracts. So this also we quantified by measuring the fluorescent intensity in different groups and different sets of experiment, and we have shown the increase in the fluorescent intensity after the treatment of the extract at different doses. This is for checking the anti-apoptotic nucleus or apoptotic nucleus by using the DAPI staining. we have seen that in control, the nucleus are intact, but in the rotenone treatment group, we can see that the disintegrated apoptotic kind of condensed kind of nucleus. And in the drug or the extract treatment groups, we observe that the number of apoptotic nucleus are decreased after the extract treatment. This also we have quantified by counting the number of apoptotic nuclei, and we see that in control there is less number of nuclei or almost negligible amount, but in rotenone treatment group there is high increase in the number of apoptotic nuclei, which is decreased after the drug treatment. So in conclusion, I want to express my observation that all the three traditionally used medicinal plant which we have included in the studies have neuroprotective property in the SHSY cell line, which is inserted with rotenone, which is demonstrated by increase in viability after the extract treatment and increase in expression of MAP2 protein in these cells after the extract treatment. And the possible mechanism of this neuroprotection is by scavenging ROS, as we have observed in the DCFD assay, and then by preventing reduction in mitochondrial membrane protection, as we have observed in the rhodamine 1,2,3 dye staining, and then by preventing apoptosis, which we have demonstrated with the DAPI staining. Thank you very much. Before that, I want to acknowledge my student, Hizam Aruna Chanu, who is the main contributor for this work, and then Yaifabi and Puza also. And I want to express my sincere thanks to Dr. Bila also, who has given us the accessibility to the fluorescent microscope facility in the life science department. Thank you very much.
Thank you, madam, for finishing in time. I'll just ask the organizers that if we have 10 more minutes, we can finish the session. So what do you want? You finish the session and then go for lunch, reduce the lunch time by 10 minutes, or we extend the lunch time for the proper timing and then reduce the session here? So it's up to you. So lunch is due. Okay. So OP4 will be done sometime later. Okay. So and then we break for lunch, and thank you, madam. Okay.
Thank you, sir.